Welcome everyone to Wednesday Night at the Lab. I'm Tom Zinnan. I work here at the UW-Madison Biotechnology Center. I also work for UW Extension, Cooperative Extension. And on behalf of those folks and our other co-organizers, Wisconsin Public Television, the Wisconsin Alumni Association, and the UW-Madison Science Alliance, thanks again for coming to Wednesday Night at the Lab. We do this every Wednesday night, 50 times a year. Tonight, it's my pleasure to introduce to you Charles Konsitzke of the Biotech Center and Danu Shanmuganayagam of Animal Science. Chuck was born in La Crosse, Wisconsin, and went to high school in Tomah. Then he went to UW-La Crosse, where he majored in finance and business administration. He's been at the UW since 2001, and he's been here at the Biotechnology Center since 2008, and he is currently the Associate Director of the Biotech Center. That means he is my boss. Be nice if you can. I can. Danu was born in Sri Lanka, in the city of Jaffna. Okay. And then went to high school in Singapore. He came to UW-Madison back in 1992 and studied bacteriology, genetics, and biochemistry for his undergraduate degrees. Then he got a PhD in nutritional biochemistry here at UW-Madison. He postdoced at the Primate Center, working on that project that shows caloric restriction lengthens life. That's why I will live to 61. He's now an assistant professor in the Department of Animal Sciences, and he's going to talk with us tonight about turning piglets into personalized avatars for sick kids. Please join me in welcoming both Chuck and Danu to Wednesday Night at the Lab.
Good evening, everybody. Thanks for coming out here. I appreciate it. So we were asked to sort of talk about how we got to creating pig avatars for personalized medicine. And so we're going to tell the story about how we got here. But before I do, I wanted to preface by saying that most of what we've done so far is a work of a lot of people. Some of my research teams out here, but it's actually a collective effort by multiple research teams, and it took a lot of people to get this shuttle launch, as the metaphor you'll hear later. Our journey really began with a baby boy who had a pigmented mark on his back side, and I'll let Chuck start that.
Thank you. So Mason is my son. He's 8 years old right now. And as Tom stated, I'm the Associate Director here at the Biotechnology Center. However, I've been facilitating and administering research for about 17 years. I've got a tact for helping very unique projects come to fruition, and I had one challenge ahead of me. So Mason was born in 2010. We had some early concerns, 5 pounds weight. He had GI issues, which required a special diet. He had jaundice. And then at 6 months old, we noticed a pigment on his rear end. And myself being who I am, I started to investigate this and come to the horror. I shouldn't say horror. What scared me is I noticed something called a cafe au lait spot in Googling it. And the worst thing to do is Google anything. So at his 9-month appointment, we talked to the pediatrician, and we stated, well, we believe our son has neurofibromatosis 1. And the pediatrician said, what's that? So right there, my alarm, being a parent, having an infant, your alarms go up. So I immediately changed my insurance company, and I transferred over to the UW Children's Hospital, where at 1 year of age, he was clinically diagnosed with an F1. So at about a year and a half, we started seeing, well, from year to a year and a half, we saw regression. There was a reduction in kind of the speech. We could tell that he was having difficulty hearing. His gait and balance was off. His behavior was changing. He was hitting his head. And so we took him in, and they did a hearing test on him, and they believed that he was having a hearing issue, and they also saw that he had a pretty substantial ear infection. So they went in, and they did an MRI prior to putting the tube in, and what they saw was a benign mass in that region of his left ear, about the size of a ping pong ball. And it was spreading into the nasal cavity as well, because what a plexiform neurofibroma is, it's not like your standard tumor. It's not like a benign tumor where it's a solid mass. This is a type of tumor. It's your nerve cells that meshes, and it grows wherever there's space. So you can imagine what my wife and I were up against. So after many MRIs, cocktails of medications where we don't know the paths of what medications, therapy, however, the therapy couldn't commence until he was diagnosed with autism first. Then the therapies could commence. So NF1 as itself wouldn't allow the doors to open to therapy. We had to get him diagnosed with autism, which is a byproduct of NF1, to open those doors to therapy. It is a lifetime worry for us because mortality can occur at any moment. So I don't step down. I grew up in a military family. I fight forward. And I told everybody I knew this disease picked the wrong person. So I started networking with other individuals with NF. It's a very small community. Started discussing issues with NF. I started looking at avenues of fundraising for NF because I'm priming myself for what the next step is. So a lot of this stuff was occurring at home, very long nights. I have my file right there. So then I did the preamble processes. Because of my position, I need to make sure that I'm not conflicted. So I spoke with leading faculty members on campus. I spoke with UW Legal. I explained the situation. I explained how I'm proceeding. And I explained how this isn't interfering my position. I got the green light, so I started moving forward. A company by the name of Pre-Scouter, which focuses on custom intelligence on demand. What they do really is they look at all the academic research that's happening in the nation, and they look for companies that would want to acquire it. So the companies actually hire Pre-Scouter to seek out these kind of interesting types of research. Well, they gave me one of their teams of postdocs for a year to investigate the bottlenecks and weaknesses in NF1 research. This occurred in 2013 and 2014. So I was receiving monthly reports. I was receiving in-depth reports on researchers internationally as well as nationally. I was looking at their capabilities, their strengths and weaknesses, and you can see the chart on the right there, as well as that's from one report, that's 1 month's report, 13 academic institutions, 3 others, 1 technology, looking at everything in depth. I started reaching out to national NF researchers, and I was asking them, what are the bottlenecks? What are the issues you are seeing ahead of you? I was reading a plethora of NF publications, manuals. And what it came down to was the model size. So NF is very unique, and we'll get into that a little bit later, but what was occurring with the model sizes with mouse and rat, which are exceptional models, don't get me wrong, they are definitely needed in research. But when a mouse would have possibly a neurofibroma tumor, as my son does, it could grow to a mass where the mouse then dies and the research can't continue because of the death of the mouse. Cardiovascular issues. You couldn't perform some of the research. This was a postdoc out of Canada. She couldn't move forward with her research on a cardiovascular issue with NF. So what the outcome was is she completely went off of NF altogether, and she went down a cardiovascular route, which was devastating. And you see this a lot, or you see this in NF research. So swine model, I communicated with a senior faculty member outside of UW, and I asked him, I said, have you ever thought of a swine model? And he said, no, but that's a great idea. So right there, I had my goal, because we're in Wisconsin. I worked on a farm. So one day I'm up at the UW Day at the Capitol, where we meet a bunch of legislators, senators, and we're kind of explaining what we do on the Capitol, kind of promoting what activities happen within the center or on campus. And after 4 or 5 hours, you're kind of like exhausted. So I was standing next to the railing, almost, and you can see my table's over there, but I was standing in the railing right there. Danu's table is right here, and Danu and I started just chatting, and we were talking about what both of us do. And then he said, well, I do biomedical research with swine. And I said, oh, I have a project for you. So I'll hand it off to Danu.
So animal models. Ultimate goal for why somebody is looking for an animal model is because they're trying to find a therapy or a cure for something. You're trying to study science, but ultimately you're looking for a cure, for a therapy, or a therapy. And if you look at what drug development for any disease is like, you sort of wonder why it even happens, because you often start out with about 5,000 to 10,000 different compounds, and you move across a drug discovery phase. You go through preclinical testing and 3 phases of clinical testing, and finally FDA approval. So from these 10,000 compounds, you're lucky if you get 1 approved. This is a very long period of time that this goes through. And drugs that reach the clinical, by the time they get to phase 2 clinical trials, the failure rate is 95%. Okay? And if you look at the cost of development, right now we're somewhere around $2.6 billion per drug. And it's fairly unsustainable. And if you look at what the cost is, the cost usually comes from the failure rate. Okay? So what's figured into the cost of any successful drug is all the other drugs that failed. And the primary cause of failure is that they didn't have the right model early on in the development phase. Because if you fail fast, fail early, you would save a lot of money. If you let it go all the way till the last minute to the clinical trials, you've already spent a lot of money, a lot of time invested at that point. Even if you could redesign the drug by a little bit, you've already committed so much effort, there's no turning back. Right? So essentially the paradigm of drug development is you're trying to get from scientific discovery to clinical therapy as a clinical impact. And the gap in between is the critical part. Okay. Most of the time, we use rodent models to try to get across this gap. And rodent models are extremely valuable. They've been very valuable in understanding a lot of disease process. However, they do have their limitation. All models have limitations, and rodent models particularly have limitations when it comes to translatability. And in the past, we've tried to sort of close the rest of the gap with primate models, but for many different reasons, including ethical reasons, practicality, and the number of drugs being developed, they just don't, they cannot sort of be that closing of that gap. Which then comes as how do you close this final gap in a way that's translational and where you can get to success with the lowest amount of expenditure of time and money? The pig. Pigs are quite amazingly very similar to us. In fact, I've studied cardiovascular disease for most of my career, and I would say from perspective of cardiovascular disease, the pigs are far more similar than our non-human primates. In fact, non-human primates rarely get cardiovascular disease. They don't die of cardiovascular disease, whereas you can easily get pigs to do that. That's because they're similar in genetics, anatomy, size, a lot of physiology, pathophysiology. But even beyond this, you can actually put them into sort of lifestyle. You can sort of model lifestyle that mimic humans. You could actually feed them very human-like diets. You can actually feed them McDonald's and they'll eat it, okay? Good luck trying to feed a Big Mac to a rodent. So, however, I mean, the value of swine models is fairly recognized. It's been recognized since the earliest of time. In fact, most of what we know about anatomy was modeled by swine, a study of swine. So why aren't they popular? Well, because swine research has a certain amount of requirements that isn't easy to get, especially nowadays. One is you got to have availability of swine models, okay? But with having swine models, you got to have expertise in swine. And that's kind of also a dying thing, especially with agriculture becoming less and less popular in agricultural research. Then those who used to have a lot of knowledge about swine have gone away, and there are new investigators coming on board with that. So you need to have understanding of swine genetics, anatomy, physiology, breeding, husbandry, behavior research, surgery techniques, so on and so forth. You also have to have the space, facilities, and supporting resources. Compared to a mouse, the swine takes a lot of space, not just physical space, but they need space in other aspects as well. And then you generally want to have a close integration with the medical research, because if you're going to use these models, you're going to need imaging technology, techniques, various imaging modality, MRI, CT, whatever it might be. So the closer you are to a medical facility that has all those, makes those studies feasible. And luckily, at UW, we have it all. Okay? And in fact, it's been sort of the very basis why I have the career I have. So I wanted to talk about, so when Chuck approached me with this is a complete switch, but my confidence of why we might be able to tackle this comes from the fact that we've had successes with swine models. One, my colleagues and I have created sort of the a miniature swine breed. This is a miniature swine that actually stays human size, allows us to do a lot of research that you won't be able to do with the sort of the conventional meat swine in the meat industry, because those animals tend to get to be 300, 400 pounds, and don't quite anymore replicate the physiology of the human anymore. Okay, they've been selected for agricultural livestock breeding. So, one of these, of all the many different sort of advantages of the breed, one of the advantages, especially when you're studying chronic diseases in the Western culture, is that it has very plastic sort of body composition, which is that if you were to feed the miniature swine more calories, they will get obese, and they will also start to develop sort of obesity associated sort of phenotypes, which is metabolic syndrome and so on and so forth. And we've been able to study those in our models. So we believe that it sort of mirrors sort of the complexity of the human physiology. One line of this miniature swine that we have used fairly extensively is one we call the FH swine. This particular line of the miniature swine has a naturally occurring mutation in its LDL receptor in the liver. Basically, this receptor is responsible for clearing cholesterol out of your or regulating your cholesterol in circulation. Okay, so it has a faulty receptor, like many people do. Those who have high cholesterol levels, a fair number of them have a faulty LDL receptor. So this swine, essentially over its age, gets high cholesterol. And like humans do, it also gets atherosclerosis, cardiovascular disease. Here's a coronary artery of the swine. It should be all open here. What you're seeing is blockage. This is plaque that's grown in here. And this is a histology of that plaque. And it has all the hallmarks of human plaque, which is you can't get this type of complexity in any other animal model. In fact, if you were to, when we've taken a vessel and sent it for a pathologist, the human pathologist, they often can't tell that we gave them a swine sample because it's indistinguishable in disease. And we've used this model now to do a lot of different studies. I'm just going to highlight a few. We've used it to develop and advance new stents, new drug-eluting stents. We've even used it to develop new imaging modalities. This is a MRI imaging technique. We were interested in whether we could use MRI imaging to detect cardiovascular plaques developing before they actually do. Can we tell before it happens? Mostly we were interested in predicting this before somebody actually develops heart disease. So we've used this model in many different contexts. The reason we could do all these studies isn't just the model, but the unique resources we have at UW, and I mentioned that earlier. And this, one aspect of it is the facilities. And I just want to, a lot of people are sort of amazed, even those who've been at UW a very long time. In fact, Chuck, when I first told him, he was sort of amazed that it even existed. He's been here a long time. He knows the research infrastructure. We have facilities. One of our breeding and sort of housing facilities houses up to 1,500 animals. Okay, it is efficiently designed by actually the chair of my department. He's been here a long time, and he designed this facility. It's so efficiently designed that it's actually run by 2 to 3 individuals. So if you think about cost of research, it doesn't get as big as you would think if you do it right. It's also what we call a SPF facility, or a specific pathogen-free facility, which means you have to shower in and shower out. It's always interesting taking collaborators in because we usually tell them ahead, you're going to have to strip down, and it's an icebreaker when first thing you do with a collaborator is you're stripping down, and then you go through the showers, and then you get shared undergarments on the other side, and but these are very unique facilities, allow us to do the kind of research we do. One of the other facilities that I manage on campus also houses a fair number of swine. I want to give you some comparison. Even some of the top-notch research universities, Johns Hopkins, for example, generally they can only handle anywhere between 3 to 10 animals at a time. Okay, and from a cost perspective, like what we call per diems, we are at about $8 a day per head per animal, whereas most of these universities who can do swine research are at about $45 to $50 a day per animal. So you can see how that works out. So, and we have fully equipped surgical suites and so on, so you can do all this sort of research. So this sort of allows us to do a lot of research. In addition, we also have our own luxury biomedical large animal transport. This was actually designed by Jamie Reichert, who's actually one of our facility managers. And it surpasses all the standards of transporting animals. It's got everything from remote telemetry to monitoring the animals by video. So the driver can actually see the animals at all times. They can monitor temperature, everything like that. Because we wanted to make sure these animals were in best condition. They were in their normal physiological state. Because we wanted to have animals that we developed that can be transported to any collaborators across the country, so they can be used by others, because we want to share what we create. So I mentioned that we have all the requirements for doing swine work, but there was one more thing that we didn't have. That was genetic engineering swine. So when Chuck approached me, we hadn't done any genetic engineering work. And so that was an undertaking. And what we realized really soon was that genetic engineering of swine is sort of like launching a shuttle. It takes teams of people working on everything equivalent to O-rings and bolts that have to get assembled, and they have to come together. And they have to come together with window of launching. Literally for gene editing of swine, it's months of work. Everything has to be perfectly timed. We have about a 4-hour window to make it work. If it doesn't happen or something goes wrong, then you're back to months and months of work. But in addition to genetic engineering of swine, NF1 is a very complex disease. So we have the complexity of genetic engineering, and then we have the complexity of the disease that we're going to study. Let me tell you a little bit about NF1. NF1 is one of the most common monogenic disorders, but you won't really hear that. If you haven't heard that of NF1 before, that's rather strange, because if you think about it, everything cystic fibrosis, muscular dystrophy, Huntington's disease, Tay-Sachs combined, NF1 is actually more prevalent than those, all combined. So it's not a rare disease, although most would treat it as a rare disease. It's caused by a mutation of the neurofibromin gene. It's a fairly large gene, one of the largest genes in the body. It affects about 1 in 3,000 birds. Okay, that's based on how we clinically diagnose it. Our recent research seems to indicate that maybe there are a lot more people with NF1 than perhaps are diagnosed. Individuals with NF1 are predisposed to both benign and malignant peripheral and central nervous system tumors. And this isn't just one tumor. This is lifetime of tumor risks. You can combat one. You just never know when the next one's coming around. Most tend to be benign, and the complexity of that is that it's the tumor size tends to impair function. If it's in the brain, obviously it's pressing against things, it's going to complicate things. But about 10% of those, where the tumor actually is malignant, it'll spread. And in those individuals it spreads, the prognosis is very poor. Absolutely, pretty much, there isn't much you can do. It almost always comes with some level of cognitive impairment. Everything from attention deficit hyperactivity disorders to autism spectrum disorders, learning disabilities, so on and so forth. It also comes with bone disorders. Some individuals are born with bone deformities or fragile bones, all sorts of sort of things. And if you are lucky enough to make it past a certain amount of age in your past your teens and so on and so forth, then you get hit with cardiovascular disorders that come in at that point. And the complexity of this disorder is that there have been over 3,000 unique NF1 mutations that have been clinically identified. Okay. And no one child is like another because of that. In fact, these phenotypes aren't very consistent. Every child has different phenotypes, different severity, depends on the mutation, probably interaction between mutation and other factors like diet, and what else that we don't understand. So we embarked on trying to create a model. We set out to create a single model. And this is sort of the schematic of how this works. But essentially, we have these embryo donors, pigs that are going to donate embryos that we genetically engineer. And a lot of months of work goes to figuring out exactly where to cut and snip the gene, how we're going to recreate a particular mutation that we're trying to model. And then and the edited embryo is then implanted back to a recipient. We do this by surgical means. A recipient swine that's going to take it and carry it to term. And like I said, we have to reproductively synchronize all these animals to these where they're 24 hours apart in their reproductive cycles. They have to be exactly that, otherwise the egg from one other will not carry over to the term. So a lot of kudos to actually Jen Nite, who's in the crowd here. She's my lab director, and that's her son who was pointing at her, who really worked a lot of this methodology out. And let me tell you, her background is in botany, so when I put this on her, she took her about 2 months to try to piece these things together. And there were a lot of other people then who actually helped carry this whole thing out. So this was a team effort. We set out to create a pig model, and in addition, I think we ended up creating much more. We created a team, we created a vision, and we created actually a community around this project. There's a lot of work that goes through this process, and we were happy to have the first litter of NF edited or piglets carrying the mutation in the NF1 gene. And one of them grew up to be Tank. He was the first genetically engineered by CRISPR on campus, and he was also the first genetically engineered NF1 boar. This is actually when he's fairly small. He grew up to be quite the tank. Actually, his name was actually quite sentimentally named by Mason, actually named him. And maybe Chuck will, if you approach him, might tell you the story behind how they came up with that name. Our central mission really is to share the models we create, because we want to get it in the hands of people who have ideas for therapies, who need this to understand various aspects of the disease. But that doesn't mean that we aren't doing our own work. Dominic, one of my graduate students, is in the crowd here, he's in the back there, and so we're also looking at mechanistic stuff. Here is some indication of that understanding the disease. So these are stem cells that we've taken from our pigs that have the mutation versus those don't. Those either have a single copy of the mutation or a double copy of the mutation. And we were looking at how do those stem cells transform to become various brain cells, and we can already see differences in how they behave. If they have the mutation, they seem to sort of speed up through their differentiation process. So we're trying to understand now, how is that affecting the phenotype? Is that causing those cancers? Is that causing the cognition? In fact, recently we were funded by the Department of Defense to develop a gene therapy. We have an idea for perhaps sort of interfering with these processes, at least in part of the patient population. So how do we get from a single model to patient-specific models? Well, our first generation of pigs, we really focused on phenotype. We were trying to pick mutations that were associated with certain phenotypes, cognition phenotypes or tumorigenic or cancer phenotypes. Then we sort of advanced to second generation of models where we were picking more types of mutation. Are there huge deletion of the gene? Are there just a mutation that completely sort of disrupts the entire gene, or some things that sort of change the wording? So for those who are not familiar with sort of the genetics, it's like your genome is like a book. Every chapter in that book is sort of, think of it as your chromosomes. And in every chapter there are passages. Those are your genes. And NF1 is one of the biggest passages in this book. And we don't really understand the message of this passage because we don't really understand the protein that encodes the neurofibromin. We don't really understand all its function yet. All we know is that sometimes all you have to change is a single word, and it changes the whole message of the passage. In some kids, they're missing a whole sentence that completely changes the passage. Others, it's some disruption that makes you read the passage differently. At the end, the story is different, and the way the story changes sometimes changes to have all lifetime of cognition problems, tumor problems, bone problems, and we don't know how to predict that quite yet. So as we wanted to set out to create a model of NF1, we realized a single model isn't just going to do it. Because every child is different, and so what if you have a single model? We're not sure which child that will help. So we came up with the idea that we had to create patient-specific mutations. And so model something for a child. And we have started this process. We've identified the clinical population to start this work with. We're waiting for IRB approval process. And that's when we sort of realized this is somewhat a new sort of area, because we weren't sure how to get IRB approval. And because we weren't sure what are we asking approval for. But as we were thinking through that, we sort of realized the dilemmas and the ethical sort of conundrums that we had to work through before we proceeded too far, which is, for example, if we were to create a pig model that models a particular mutation of a child and if the pig dies, what do we do? What is our obligation? Do we inform the parents? But maybe the pig dies, but maybe the child wouldn't. So all of these had to be we had to work it out, and we haven't worked all of it out. We've worked some of it out, and we're trying to move forward. But our long-term goal is that we'll have a pig for every child. And what that does really for NF1 is that right now, the way a drug for a child is figured out is you try a random set of drugs. There is no schematic for it. So that means every child sort of has to go through all the side effects of all these drugs. And usually they're on a cocktail of drugs, one for cognition, one for this or that, and the side effects of that. And if they're not lucky enough to find the one that sort of works right away, there's time ticking away. And we're talking about tumors that are growing. And they're losing time in terms of cognition issues. And they're getting delayed in learning and so on and so forth. So what we're hoping is that when we do this, we will use the pig as a way to screen, the drugs. One is to also start to use the different pigs with different mutations as a way to sort of try to sort of dissect out why is a mutation here causing this phenotype or so on and so forth. Is it interacting with diet? Because we can put diet in pigs. We can put them on a high-fat diet. We can put them on a high carbohydrate diet and see whether that influences it, because we don't understand the dietary influence on this disease. In fact, it's interesting. If you talk to clinicians, they might say, well, there's no data on this. If you talk to parents, they'll say, oh, my kid responds to, I don't give my kid Skittles or this or that because something about that sort of makes their symptoms worse. They act out. But we don't know because we haven't really studied. We don't have the models to study. And we're hoping this will allow us to study it. As we got this idea, we realized we could use this paradigm for many other things, including cardiovascular disease, which is also fairly complex. Perhaps not as complex as neurofibromatosis, but it is one of those diseases with multiple things acting on it where everybody is different. This idea of precision medicine. We also, that led us to gene editing ideas, and in fact we have an initiative going to create organs that are tailored, pig organs that are tailored and compatible with a particular patient requiring organ transplant. That's a whole different area, but I can tell you that, again, we're really hopeful about that too, because right now there's about 5 to 8 year wait for an organ. It's about 5 years in Wisconsin, about 8 years elsewhere for, like, say, a kidney, and about a fair number of that population will not make it because there aren't enough organs to go around. So, in fact, if you can custom tailor organs, and pig organs being very close to humans with just the right enough things to fool the immune system, we believe that's going to open the door. We are very lucky. I feel very lucky to be at UW, to have the resources, having worked with animal models, because the right animal model is a big game changer when you talk about diseases. And in fact, historically, animal models have made all the difference. And if you're lucky enough to have a relevant model, and if you have the resources to do, like, pig models, for example, then you can have a great impact. And so far in a lot of the other diseases that we've studied, I've been very lucky to see a lot of things that we worked on go to clinic, clinical trials, go into clinic and be used in clinic. And we're really hoping that we can do the same for NF1. Thank you. Yes.
So does NF1 have an effect on reproduction in humans and in your pigs?
We have not seen it in our pigs yet. It's fairly early. In humans, there are subtle reports here and there. It's kind of hard to figure out whether it's NF1 directly or the consequences of some other things that come along with it.
So you hope that from one pig you can...
Yeah, we've already gone through several generations, and so that's sort of we're working well so far. Yeah. Yes.
Well, thank you so much for your presentation. The different disease, the question. So I have a 25-year-old daughter who was diagnosed with type 1 diabetes age 3. And, you know, what you described with the mouse model, and, you know, I think type 1's been cured, the mouse model, 50 different ways, and the whole pipeline challenge, all that rings so true. And, you know, we had told her at age 3 that in 10 years she'd be able to have the operation that would fix it, and, you know, we're 22 years later, it hadn't happened. But there's research in, so my question is this. I think there's a group in Australia that has altered, and I'm not a biologist, has altered the pig beta cell that makes the insulin to be very, I think, similar to a human beta cell, so it's not rejected quite as much. You still have to encapsulate it or something like that. So my question is, are you aware of that work to humanize or make that beta cell more similar to humans? And has there been any work here at the UW along that line of, you know, beta cells or islet cells?
Yeah, there is a lot of islet cell work going on in the human line or regenerative medicine area. It's coincidentally that you would ask this question. In fact, we were just funded 2 weeks ago to actually engineer swine, to engineer islet cells, so swine, for this exact purpose. It was a collaboration between my group and a group over at the transplant surgery. So we're going to do that on the swine end, but there is also work going on with islet cells, in human islet cell work as well, on campus, yes. Yes?
Do you have any initial results on using these patient-specific pigs that have been helpful for the patients?
No, we are still very early in the process. In fact, we haven't modeled a particular patient yet. We're in the midst of it. So, and it is a initially, it's going to be a little bit of a longer period because you have to actually characterize. We have to go through the characterization. We have to make sure. The pig has been a great model, but I don't want to. As a scientist, I can't. That's just theory, till we actually prove it, right? For every condition. So we believe it will be for NF1 as well, from all aspects of what we looked at and so far. But we have to actually prove that it is indeed true. So we have to do this sort of characterization and validation stage. So that's where we are right now. Yeah.
I think you mentioned there were 3,000 mutations for NF.
Mm-hmm.
Is that an average for other genes? Are there that many mutations, or is that unusual?
It's fairly unusual. It is a big gene, so and 3,000 is probably a low number. It's 3,000 from people who are sequenced, genetically sequenced. A lot of NF1 patients are not genetically sequenced because it's not covered by insurance. So, in fact, we were actually both approached by someone who his wife was recently diagnosed with NF2, which is fairly similar, when she was 60. And they have kids who are in their 20s. And so he was wondering, he would like to get his kids sequenced to know whether they carry the same issue. And he said, we don't know how to help. I mean, the cost of sequencing is not that high, but unfortunately the healthcare system, it's unaffordable for most people to do that. So yeah, chances are there are a lot more mutations than the 3,000. Those are, yeah.
Can you just pick certain genes then to sequence for these people, and they only pay for that?
Yeah, certainly you can do that.
So we're talking probably hundreds of dollars to have that sequenced.
That would be the actual cost, but the cost of actually doing it from a healthcare perspective,
But I mean for a person to pay for it themselves, they would be hundreds.
Well, the thing is that there is no way to have it sequenced for them because of all the limitation. I mean, they can't just walk into let's say the biotech center and ask to be. They can now, right? Yeah, but because of the how you manage the information as well, right? So yeah.
I guess what I'm getting at is I had I think it was 9 or 11 gene sequence for breast cancer.
Yep. Yeah.
And I think it costs $420.
Sure. Yeah. So I don't know whether it's NF1 on the panel yet or no. Yeah. So some of those. Yeah. So. Yeah, actually, we've been talking to a few companies trying to get that on the panel, so it's a standard sequencing as well. Yeah.
Thank you.
Sam, did you have a question or something?
I did not.
Okay. Yeah.
So during your presentation, you talked about the difference in the different models. Using mice, using swines, using chimpanzees or whatever. So. Are there other large research universities in the United States who are also on this path of using the swine model? I'm thinking, maybe the University of Arkansas or maybe Iowa, something like that. Just to see, are there other institutions that have made really good advances that you guys can partner with and maybe accelerate some of the ecosystem around all this?
Yeah, there are a handful of them, meaning about maybe 3 or 4. And most of them were all the big ag universities, kind of like us. Missouri, University of Illinois, Champaign-Urbana, Iowa. We are actually partnering up with Illinois, actually, to develop other cancer models. The issue with most of them, so there are a lot of these universities can develop models. Unfortunately, they can't all use that model once they develop it because they don't have a medical school or they don't have the imaging capabilities that we do. So we have actually dedicated imaging for MRI, CT, PET, ultrasound, all clinical standard equipment. In fact, unlocked equipment. So some of those imaging I showed you there, it's because those were algorithms actually developed by our radiology and medical physics group. So we have systems where we can actually custom design the imaging modalities. So they don't have access to that. So they send out just pigs to other people who need it, which isn't quite the same as do it under your own, under the same roof. But yes, we are as much as we can trying to partner up and collaborate. But funding is right now is the biggest to some of these collaboration increases the kind of funding and stuff like that you require. Yeah. Yes.
How is NF1 diagnosed, and how often do you think it's missed?
You want to talk about it, and I'll give you. I'll talk about our data as well. You want to talk about the diagnosis right now?
So diagnosis is as well as reported in the report. It's 1 in 3,000 births. However, I mean, we're seeing that change because that's only looking at the clinically diagnosed, and that's typically by phenotype, well, clinically, but then also phenotype. So they don't really have some of these individuals don't really have a true diagnosis. It's just by phenotype, the visual.
So how many cafe au lait spot?
6.
So if you have under 6 cafe au lait spot, in fact, I have fairly young children, and when we started seeing cafe au lait looking spots, of course, and then you start counting because it starts increasing in number, and you have to get this threshold. So it's sort of a subjective sort of measure.
But some of the latest publications that are coming out now, you're seeing that actual 1 in 800. I think it's in Israel. The last 1 in 800 because they're clinically sequencing their infants. So that's the catch here. One of the things that I didn't speak about, and one thing that Danu didn't speak about, is in our community that we have here, there are parents who really didn't even know that they had NF1. And it wasn't that bad for them. They may have had a cognitive issue or a slight behavioral issue, but then when they had children, that became extremely aggressive, and their children has major issues, major GI issues, where they have to be tube fed, tumor issues, where tumors have to be removed from their brain, etc. So one of the things that I'm also working on, because I've got a colleague who leads, she's one of the leading individuals in infant screening. I'm hoping to see NF1 be put on that screen because I truly believe it's probably maybe 1 in 1,500 or 1 in 2,000 because we're not diagnosing. However, with the evolution of sequencing occurring, platforms are sequencing much more data now that we're seeing a reduction in sequencing costs where maybe in 5 years from now, every child will be sequenced and we'll have a different answer then.
Yeah, we have some data coming from our swine work that some mutations may actually stay fairly silent. Some mutations of the NF1 gene might actually stay fairly silent. In fact, there's an interesting paper that came out a few years ago that showed when they sequenced the breast tumors of women with breast cancer or whatever the tumors, 1/3 of those tumors had actually a mutation of the NF1 gene. So meaning NF1 gene might be actually involved in breast cancer as well. But interestingly, that paper, they basically glossed over the NF1 gene mutation because they didn't really know what NF1 does or anything like that. Yeah.
Could you speak briefly on dominance recessive of the different alleles for NF1?
Yeah, it's a really interesting NF1. That's another whole mystery. So most individuals, as far as we know, the data is that you're only born with a single copy of the mutation. In fact, 2 copies of the mutation is lethal. We have some data that it actually might not be depending on where the mutation is. And usually you need 2 mutations to get a tumor phenotype. So that means people are born with 1 copy, 1 mutation. They're acquiring the second mutation after they're born. And we're not sure how that's happening because it can be coincidence that the second mutation is happening the same gene again unless the first mutation somehow influences the second mutation.
Same codon or whatever is getting.
Yeah, either the same or it's in the same gene but a slightly different mutation is happening. So it's like that first mutation is driving second perhaps. that's why the model now becomes interesting because we can start studying some of these things.
One other interesting note is one thing that we're witnessing is we believe that the gene is possibly a scaffolding gene where other mutations that occur throughout the genome are influencing it differently, because we have cases where in the patient world where there's a family that has the same mutation but each child has a different outcome.
Yeah. I saw some hands up. Yes.
I had a question on the, is the time frame the same for the observable issues with NF1 in both human and swine, or is there an accelerated time frame for the swine?
So we don't have a time frame established for a swine yet. It's early in the game to sort of establish that. In humans, the various different symptoms have different time frames. And there's also a sex-specific sort of expression of symptoms. For example, optic gliomas, so these are sort of tumors of the optic nerve, seems to be more prevalent in girls than in boys. And it seems to be, actually one of our collaborators has got data showing that it seems to be that small little spikes of estrogen. These are pre-pubertal girls, but small levels of estrogen signaling seems to be driving that tumor. And there are certain tumors that come in much later in life, after puberty or so on and so forth. So these time courses for the different phenotypes are different.
I had a second question.
Yeah.
Do any infections seem to be a precursor before these observable issues of NF1 come up?
Not that I know of. Not that I've read about or reported either. Yeah. Yes.
Are there certain ancestries or demographics that would make someone more likely to have an NF1 mutation?
Have not seen that, but then again, we have not done extensive studies.
Except the publication that from the history. You should actually speak about.
Yeah, it's interesting. Historically, NF1 has been around a long time. There's reports as early as 1500 BC of NF1. So this isn't something new. And it's generally misdiagnosed, has been misdiagnosed for other things that have very similar stuff. But is it one particular ancestry? I don't know, but it's certainly been around a long time. In fact, there is a MRI, CT scan of a mummy, and that shows that there was a plexiform neurofibroma, and there's fairly good evidence that it was actually an NF1 mutation that caused it. Yeah.
Sir, is it more prevalent among Jewish folks? Is that why they're screening for it and sequencing of Israel?
I think Israel is just they're just doing gene screening.
No matter what. It's
yeah.
Just, yeah.
I'm wondering about the cafe au lait spots. Is that a red flag for diagnosis? Because we have 2 Amerasian grandchildren, and we discovered that Asian people have cafe au lait spots.
Mm-hmm.
So
Yes, they do. My father-in-law is from Hawaii, who came from the Asian descent. So if you look at some of my nieces and nephews, they have cafe au lait spots. However, they only have 1 or 2, and that's not an issue. When you see 6 or more and the size is larger than a centimeter, that's the precursor for NF1.
And we're not entirely sure why that's the case or so, but it seems to be that they're also increasing in number as well, right? Yeah.
Yes. Yeah, you'll see freckling. There are other things that will appear later in time as well.
how effective is the treatment?
There is no cure. There's no treatment.
Well, so the drugs are just keep working on the symptoms?
Yeah, most of them are working on symptoms, and then others are just basically trying to control the tumor size.
The tumor size, yeah.
Yes.
Are the 5 or 6 cafe au lait spots, do they tend to happen at the same age in all children?
No.
No.
You can see, well, regarding my son, his first one was on his butt, and we thought, oh my, it's a beautiful little birthmark. And then another one appeared, and then another one, and they're continually appearing to this day. So you're in denial sometimes. I am, I should say. But yeah, it's
Different ages.
It just continues, over time.
And not everyone also displays cafe au lait spots, which makes it like diagnosis more difficult because there are many NFN cases without cafe au lait.
Well, and Danu stated too, diagnosis. One of the things, our pediatrician at 9 months of age said, "What is neurofibromatosis?" You see that throughout the nation. Many physicians don't even know what NF1 is. So if somebody comes in with a behavioral issue, oh, it's just behavioral, let's give him these meds. Or it's cardiovascular, let's give him these meds. When actually it's NF1. It's not, NF1 is an umbrella of many, clinical outcomes. And if a physician doesn't know that, they're treating it as one thing.
But is that the only way to treat it, moving from symptom to symptom with therapies that are appropriate to those symptoms even without understanding the origin?
Yeah, pretty much. Yeah. It's sort of the way it's going so far. Right now. Yeah. And one of the things that they've focused on, obviously, is on the tumor symptom, right? That's because it's sort of viewed as the harshest. However, now there was a call. We were at one of the meetings, and they said, so one of the things that a lot of the individuals with NF1 can get is these neurofibromas, cutaneous neurofibromas. Yeah. So these are like almost boiled, like they're tumor growths, but some are just covered in it. Absolutely. And some they deform you because they grow to such size. So one of the things, so those haven't, you don't pay a lot of attention to because those are not considered necessarily, they're considered benign and, not to focus on. But the suicide rate at teenage is high in individuals with this because obviously, it's a deformity. So there's been a lot more focus recently to say, can we also focus on research on that, even though it may seem cosmetic, but it has a huge impact on the patients.
Yeah, I'd say don't go home and Google NF1 because you'll be probably traumatized because the deformities are substantial.
And a lot of bone abnormalities, scoliosis is very common, and some of those are very severe, and especially those where the tumor grows to completely offset the body. It'll deform the entire face, one side.
Mm-hmm. Well, at our 3-year oncology appointment, or neurocutaneous oncology appointment, when they saw the tumor, it was growing. And knock on wood, it stopped around 3.5. But they said, take pictures because his face will probably be deformed. So you'll notice we probably have thousands of pictures of our kids because you never know, it could turn right back on. So.
Yes.
Is there any research on? I read recently that one of the things that isn't happening in the United States is any kind of analysis of stillborn children. Mm-hmm. And so the question becomes any kind of DNA analysis or any kinds of autopsies that might show that NF1 is, the cause of stillbirths or any other kinds of issues that.
Right. So this is another project we were doing is kind of related to NF1. So congenital heart failures or heart defects at birth, NF1 is one of the genes that are responsible for it. We're actually working with a number of physician groups to actually set up a training center to deal with how do you deal with the kids, because it's, if a kid's born with a congenital heart defect, there aren't enough trained people to deal with it because they're missing part of the heart, whatever it is, and NF1, that's one of the things that NF1 apparently seems to affect in development. So we're actually trying to develop pig models so that there's training as actually a model for training physicians. Because one of the biggest problems, apparently, if you want to be very good at dealing with congenital heart defects in kids, you got to have a lot of experience. It takes about 10 years for a physician to get expertise and his team to. Apparently, after 5 years, most of them give up because the outcome is just terrible, and they just burn out. So we're actually focusing on it, and the way we're trying to create the pig model is actually with NF1.
So is there any specific age where NF would be most common or most deadly?
Mortality rate is at any age. So no, I mean I know of a teenager that passed away just last year. I know of kiddos where they have to have amputations. It's at any age.
One thing I want to point out is that, so that is overt mortality, but there is also, one of the things we did mention is there's chronic pain involved. These are nerve tumors. Yep. In fact, we met I attend some of the events. That's why I said we built a community around it to get involved in the fundraising and all sorts of other events. We were at an event in Vegas where there was a concert for this, and I met a young lady. She's about 22 or so, who was going to have her leg amputated, but she was looking forward to it because she had been living with a pain in that leg for a very long time, that she just wanted the pain gone. And that was like the week before she had it amputated.
Yeah. Another issue with neurofibroma or plexiform neurofibromas is it is a nerve tumor. So if they operate, it could be as painful after the operation because you're damaging nerves than it is before. So it's really taking a risk. I mean, at first when they were talking to us about my son about one, his tumor is inoperable, but they could just tax it with a number of radiation therapies, etc., that the outcome could be even worse. And this is the case that we're discussing with the gentleman that Danu spoke about with NF2.
Yeah, his wife had she's about 60, had that tumor removed, and now she's lost over I think a month. She lost her hearing. She's in chronic pain. Her whole face is in pain, and so he not only wanted to see whether we had any solutions for it. In fact, he's in the science realm, so he can't stop his mind from trying to. He's in the regenerative medicine too, so he's thinking about how can he use stem cells and whether we would help him develop some of these ideas he has to try to treat his wife. Because if they can repair that nerve, then she would at least get over that pain that's going on. Yes.
So, just referring back to the pig models, then, and on the issue you're just talking about, how do you tell if a pig is experiencing a certain type of pain in a certain place?
Fairly difficult to do, obviously, as any animal models are, because you're not able to sort of communicate. It's very neuropathic pain. This is what it sort of amounts to. However, there's been a fair amount of pain and distress scoring done in pigs. Pigs are their cognitive abilities are actually considered now the next best thing to non-human primates. They're actually, in fact, we are setting up cognition studies in pigs. In fact, I have a colleague who had trained pigs to play video games. Yeah, and they're extremely smart animals. In fact, they're easier to train than dogs. So they also, therefore, you can develop tests to sort of assess a lot of these things. But of course, there is difficulty. I mean, even in human population, trying to assess pain is one of the most difficult things. Yeah.
Give us a thumbnail sketch of quantity?
Very little.
Very little in general, right? In any work. NF1, there is no, NIH has no funding mechanism for it.
It's classified as a rare disease. That's why it falls underneath the rare disease umbrella.
Even though it's not. And then the only funding is available is through Department of Defense's medical program, and that's from actually some of the foundations that we're involved with. It was from their lobbying efforts over the last 20, 30 years that even a funding string there. But generally, what we do, we spend a good portion of our time doing the research work. The rest of the time, we are trying to come up with novel ideas, not for therapies, but novel ideas for how to fund things.
We're running in about, 3 weeks, 4 weeks?
Yeah, and even trying to develop a business plan, if we have to spin off a company, whatever it is to get the work funded. We spend a fair amount of time to figure out how to create a sustainable funding model just as much as we try to figure out how to deal with the disease.
Chuck, you said NIH doesn't cover it because it's a...
Well, they cover it but it's underneath the rare disease.
Even though it's not a rare disease.
Correct. But it's underneath that umbrella.
So who do you talk to get them to adjust the classification?
It's been discussed many, many years, and it just, I don't think it will happen until sequencing ramps up even further and they see that ratio or the prevalence is, yeah.
And so I think one of the things we should point out is that most of this funding was grassroots. I mean, we took the risk very early on. I think you started with a golf outing, which we now do every summer to raise funding, and every other events we run races and marathons, whatever it is. We have a whole bunch of people doing this sort of thing, but it was a fairly risky thing because we basically
they took the risk because it was a minute of money. I mean, the first year and a half, I went and presented out in Vegas to the NF network saying, we've got this idea. I asked for seed money. They gifted us $50,000. I'll give you a scale. $50,000 pays maybe just one grad student. Other people who I had communicated my outcomes to, who then, they ran along on that path, were awarded $1 million. So you can see, however, there's positives to this, though.
Yeah, so, I mean, one of the things I had to say is that, you asked me about who else is doing swine model development. So the biggest swine model development are currently by company. There are like 2 or 3 companies that do genetic engineering on swine.
For-profit companies.
For-profit companies, yeah. So, which essentially, one of the advantages of what we've done with the shoestring budget, one advantage is that, not that I want to stay this way for long, but is that you get very, very efficient at it. In fact, the efficiency that we are at of genetic engineering is at par or surpasses those who've been at it of genetic engineering for a long time, like some even up to 30 years. So we did this in about 2 to 3 years. So that's one. Otherwise, we wouldn't have even thought about patient-specific modeling. We thought the other thing is that from a commercial perspective, patient-specific modeling doesn't really scale. So because why would you just create one pig, all that money to create one pig for one patient? It only works from our setting, from an academic perspective, where we're not trying to look for our profit out of it. So there is that advantage to having that other outlook to it. But I'm okay with the shoestring budget. I'm okay with that.
He likes running with me and golfing with me, so.
No, I mean, I think the funding limitations, they are a hurdle to innovation where you're sort of bogged down by not taking the leap of faith with some ideas that you have that might be a therapy simply because your budget doesn't allow it. So that's the downside to having funding limitations, that you become very risk averse. Yeah.
And I just want to make one more compliment before we're done. I just want to thank Danu and his team, because if you look at research in general, especially a population that is afflicted with a very aggressive disease, many patients never meet the researchers who actually do the research. Their team actually participates and runs with the community. They golf with the community. They go to concerts with the community to focus specifically on raising funds to help move this forward. So it's not, they're just not obtaining the money. They're actually participating with it. So I just wanted to thank you all, because I could start crying right now, but I won't, because it means a lot to this very small community. And it is a very small community. So, thank you. Now, can I just have everybody who works in research of NF1 please stand up?